I think we're fighting some good weather today, so I appreciate all of you coming to listen to the talk. There's a sign-in sheet that is going around. As always, please remember just to sign in. It helps us justify the pizza. So today I'm pleased to introduce our speaker. We have David Lubensky, who is Associate Professor in the Department of Physics.
So thank you all for coming on such a beautiful day. So when Marcy wrote me to ask me to do this, my first reply was that I don't really do tools and technology. And she said, oh, that's fine, just give a talk about what you do. So this is a talk about what I do with a few added things about the more implementation and computational aspects of it, but that's not fundamentally what my lab is good at or what we focus on. So this is primarily a talk about our research, and which is about modeling animal development. And so, okay, I wonder if we can arrange this so I can make it to my laptop without blocking the projector. Okay, that's better. All right. So specifically, we're interested in animal development. And so the fundamental question of animal development is, of course, that we have this egg here. And this egg knows perfectly well what it wants to be when it grows up. But at least to me, it's not remotely obvious from looking at this whether this is a chameleon or a chicken or a fish or what. As it turns out, it's a chicken. But it starts like this and it ends up like this, and these don't look the same at all. And so the basic question we're interested in is, how does it know to get from there to there? And from my perspective as a physicist, what this is a problem of self-organization. That is, there is, of course, a genome inside the egg that tells it that it's a chicken, but the genome is not coding in sort of a blueprint-like way for exactly where every piece of the chicken goes. It doesn't say the eye goes there and there's a certain distance to the beak and so on. It, as most of you know, encodes signaling pathways and ways in which cells will interact with each other that will let them organize themselves into the chicken in a very elaborate stepwise fashion. And so what we're interested in is, how does this self-organization happen? What makes it work well? So this is, of course, an incredibly vast problem that has occupied many billion people for over a century. And I don't claim to have general answers. What I would like to tell you about is some specific examples, and today's main example is going to be about the arrangement of cells in the fish retinal epithelium. And so here's a picture of those cells, and I will come back to what this means. But so essentially, I want to tell you today a little bit about the background of the retinal comosaic in fish, and also about epithelial sheets. So I should ask, most of you are bioinformatics grad students. Is that an accurate statement? So this means that some of you have a lot of biology background and some of you have fairly little biology background. Okay. I will leave in the biology background, and apologies to those of you who already know it. So I tell you about epithelial sheets and about the comosaic in fish, which presumably almost nobody here knows about. And so specifically the question of how it precisely controls cell fate. Then I will tell you a little bit more about the background of how this happens, in particular why we think that the emergence of a very ordered packing of cells is related to anisotropic mechanical stresses. Then I will tell you about the models we use to study this, which might be called active foam models. The more technical term is Vertex models. And tell you a little bit about how rotational symmetry breaking couples to mechanical stresses. I've added in a bit about how we actually implement these things. And then about predictions, and back to some experiments about the fish. And I think that's probably as far as I will get, although there's more material if we're going fast. All right. So we're all starting at the same place. Let's start with, what is an epithelium? So an epithelium is a sheet of cells, very often, though not always, one cell thick, and connected so that it forms a continuous 2D sheet with mechanical integrity. And epithelia are, of course, all over the place. So your skin is an epithelium, the lining of your gut, the lining of your lungs, the lining of your esophagus, so forth and so on. Anything that sort of looks like a barrier or a sheet in your body is probably an epithelium. And so these are really one of the basic building blocks of all animal tissue, of all animal organs and all animal body plans. And they come, and so, right. And so this is a blow-up of 3 cells in epithelium. And for my purposes, important features are that the cells are joined together in a fairly narrow band near their top, so their apical surface. So it's quite close to 2D in many ways. Certainly by the standards of biology, it's fairly close to 2D. And particularly, well, there are some adhesions down here, but they're thought to be weaker and not predominant for most of the problems I'll be talking about. There's very strong adhesion around here near the top. And also, behind the band of adhesion molecules in green, there's a band of actin and myosin inside the cell, of cortical actin and myosin. And many of you will know that actin and myosin are contractile. This is what makes your muscles contract, but the same filaments and motor proteins also exist in cells. And so there's this red band that's sort of like a rubber band behind the adhesion junctions in every cell, which means that these things are under tension. There's something constantly trying to contract them. And this is what's going to make it look more or less like a foam. And okay, and for my purposes, I will from now on view this as essentially two-dimensional for most purposes. All right. So epithelia come in a variety of shapes and sizes. So these are just some examples of epithelial cells in different animals and different organs. This is a fairly mature, close to the end of development, fly eye. And so there's going to be a little, fly eye is a compound eye, there's going to be a little lens in front of every one of these structures. And you can see that, these cells clearly, they're not put down at random, right? These cells that make the half moon had to know they were a half moon, and these ones here had to know that there should be 4 of them to be arranged that way, and so on. And they're arranged in a different way from this, which is a fly wing, where a lot of the cells are relatively disordered, but this is going to be a vein, and you can see that these cells are doing something different, right? They've upregulated a particular molecule along those junctions, but they've also made a very straight interface there. And this in turn is different from, this is the mouse inner ear. These are going to be the hair cells that detect sound. And these need to be arranged in a particular way. So the point is that these are sheets of cells, but they're sheets of cells with particular structures and particular arrangements of cells. And these things have functional consequences. You need to get the hair cells nicely spaced so that they can detect all frequencies of sound, and otherwise your ear doesn't work so well. And so you had to get these cells arranged correctly at some point during development. And so you might ask, how does biology regulate this? How do the cells know what shape they're supposed to be and where they're supposed to go? And that is what my example today is trying to look at. So the example is the fish eye. So this is, of course, a zebrafish, and it has an eye, 2 of them, in fact, but we'll focus on this one. And so if you were to dissect the eye and make a cut right through the center so that this here is the pupil, the light is coming in here, this is the lens, and then it's getting the lens is focusing light onto this tissue, which is more or less hemispherical. In fact, without artifacts from fixation, it's very close to exactly hemispherical. And this is the retina. And this is where all of the photoreceptors live that are going to detect the light. And so if you were to now cut out a little piece of this epithelial sheet, the retinal epithelium, and look down onto it from above, what you would see is this. And so what's notable about this is you'll see that there are alternating purple and blue cells, and they're really always alternating one and the other. So what does that mean? So some of you will know that the way we see colors is we have cone photoreceptors that are sensitive to 3 different, or have peak sensitivity at 3 different wavelengths in red, green, and blue. And so the way you decide what color something is you look at how much intensity you're getting in these 3 channels, and you compare them, and you say, oh, it's between the green and the blue, and so it's yellow or whatever. Fish works the same way, except that it turns out that they have 4 different color photoreceptors, or photoreceptors with 4 different spectral sensitivities, which are red, green, blue, and something in the near UV, so somewhat above what we can see, but the fish can see it. And so what this false coloring is that the blue is a transgene, fluorescent transgenes that's marking blue photoreceptors, and the magenta is marking the UV photoreceptors. And so what this means is that this fish has managed to get the cell fates in its retina really perfectly regularly arranged so that it always alternates blue, magenta, or blue UV, blue UV, blue UV. Moreover, if you look closely, you'll see that the unlabeled cells here, and the reason they're unlabeled is not that we don't know what they are, but just that it's hard to label 4 things in one picture. And you'll see that there are always 2 cells in between, and these are systematically a red on either side of the blue and a green on either side of the UV. So in the end, what in cartoon form, the way the photoreceptors are arranged in the fish retina is like this. So this is really a regular crystalline lattice. It's repeating the same unit again and again. And this is extremely unusual. This other vertebrates don't have this. We don't have this. Birds don't have this. Only fish have it. Essentially, all bony fish, however, have some form of lattice. And there are very few other cases. In fact, I would say I do not know of any other cases in vertebrates where the fate and position and arrangement of every single cell is controlled this tightly without any room for slop. There are cases of regular patterns, but usually there are some, there's the number of cells in between, say, the hair cells is somewhat variable. It's not as tightly controlled as this, where every cell has a particular fate and is in the right place. All right. So this raises the obvious question, how did it make that? Because after all, it started out as an egg, as we said, and the egg didn't have an eye, and it certainly didn't have a retina with a crystalline mosaic of cone photoreceptors. So it had to know to make this. And so that actually involves multiple steps, and in keeping with the general theme that biology is complicated and I'm not going to explain everything, we're going to focus on one of these steps. And so really to get a regular packing, you need 2 things. So you need, on the one hand, actually let me back up. So you need, on the one hand, you need to get the right number of cells of the right colors. So you need 2 reds for every blue, and no extras and none missing. This is the part we really don't understand well. The other thing you need is you then need to position these cells correctly in space. So they need to be in a rectangular lattice, and that's not naturally what they want to be. If you just randomly throw down cells on a surface, they will on average be hexagonal. So arranging themselves with rectangular symmetry is non-trivial. And in particular, if you look at the way these cells are packed, and I should say there are going to be a lot of pictures with yellow cell outlines here. This is always visualizing a protein called zonulocludins that sits at the junctions at the top of the epithelial cells that I told you about. Okay, and so what is apparent is that the predominant feature of the way these cells are arranged is that they're arranged in these very straight regular columns. And then there's also correlations in this direction, but those correlations are somewhat weaker. And so these straight columns are really the thing that comes out first. And so if you want to know how does the fish retina arrange itself into a crystalline lattice, one thing you need to understand is how do you go from a disordered arrangement of cells into cells that are all nicely lined up in straight lines. And yeah, that is what we want to do. Okay, so what do we know about how this happens? So first thing is that one of the very nice things about fish, indeed one of the reasons in particular fish eye is a system that gets studied a fair bit in a biomedical context, is that fish have neurogenesis throughout life. So I'm not a fisherman, but maybe some of you are fishermen, and you will know that fish never stop growing. So, they stop growing when they get killed for some reason, but otherwise they actually continue growing indefinitely, unlike us. We have a definite size and we stop. This is why you have sort of long tails in the fish size distribution, and you can have stories about the big one that got away. But because they never stop growing, their eye also never stops growing, and that means it keeps on making neurons. And the way it does this is, so as I said, the retina is more or less a hemisphere. And at the edge of this hemisphere, there is a zone of proliferative precursor cells that are dividing and renewing themselves. And every so often, some of them decide to stop dividing, exit the cell cycle, and start differentiating into cone photoreceptor cells. And so what that means is that I have a disordered packing of cells here, and then cells are going to leave this packing and sort of attach themselves to the ordered pattern and attach themselves in a stepwise fashion. So the pattern grows like this. It adds columns to the right in this picture. All right. And that's already suggestive of being helpful in making a very ordered pattern, because in general it's easier to make regular lattices one layer at a time than it is to all at once have everybody agree to rearrange themselves. In fact, fact, this is how you grow clean crystals if you need a clean silicon crystal for whatever electronic device you're making, right? You grow it layer by layer. However, that's not necessarily enough. So a few other observations. So this is a picture of the cell outlines right at the boundary between the mature cone mosaic, where you can see these clear vertical lines of cone photoreceptors, and out here, the germinal zone with the proliferative precursors. And so what you can see is that there really is a change here. So this is more or less typical of what a proliferative epithelium looks like. If all of the cells are growing and dividing, their sizes are variable, and they tend to make a fairly disordered packing. But then they, at some point in the fish, they sharply transition from there to, well, I'm going to make nice straight lines. So the other important observation is that fish have a larval stage. This is like a tadpole in a frog, only it's a tadpole for fish. And the larva has an eye, and the eye has the same kind of photoreceptors, and it grows in the same progressive way from a germinal zone. But the packing of the cone cells looks like this. And this, incidentally, is not far from what your own eye looks like. So you can see that this is not a regular, really crystalline lattice. There is some short-range order. You don't have large clumps of blue cells together. There's some characteristic spacing between the blues, and you tend to have UVs next to blues. But it's also not really ordered in the way the adult is. And similarly, if you just look at the outlines of the cell packing, you can see a difference between the margin here, which is a bit more of a mess than over here, where the cells are differentiated, but not as pronounced a difference. So what this means is that this progressive growth alone is not enough to make a proper pattern. And you have to ask what is. Okay, this, I think none of you care about Q space unless there are stealth physicists in the audience. No. Okay, so, right. So this is the larval eye. This is the adult eye. And what we want to know is what's different that makes the cells organize in a really regular lattice in this one, not in this one. And one thing we realized is that there are some changes in eye anatomy that happen round about the time that you go from a young disordered eye to a very nice adult cone mosaic. And in particular, there's this thing called the annular ligament that appears. And the annular ligament is essentially a ring of fairly rigid tissue around the outside of the retinal hemisphere. Incidentally, this is an anatomical feature specific to fish. Mammals don't have it. And it's classically said to provide mechanical support to the eye, whatever exactly that might mean. But what's clear is that if you go from having a floppy hemisphere of tissue whose edges are unconstrained to the same sheet that now has its ends pinned down onto sort of a rigid metal ring, say, you're changing the mechanical stresses on this thing. And doubly so because the retina is actually a little bit like a balloon, in that the inside of the eye is under fairly substantial positive pressure relative to the outside world. And so you have this thin sheet, and you're sort of blowing it up. You're putting pressure inside it, and at the same time you're holding onto it tightly with this solid ring. And so that should induce anisotropic mechanical stretches. There should be more pulling in one direction than the other. And our idea was that maybe this can contribute to lining up the cells nicely into columns. So the idea would be that with isotropic stresses, no ligament, you get something like this, not very ordered. But with anisotropic stresses, sort of the cells have a clear signal that tells them the columns go this way, and so they know how to make themselves into columns. So this is work done in collaboration, I should have said at the beginning and we'll say at the end, with a lab here at U of M, Pamela Raymond's lab, who actually do experiments on fish. But we are entirely computational modelers, and so our way of getting at whether this was at least plausible and what we should look for as signs of it was to go build a computational model. And so let me tell you a little bit about how we build models of these packings of cells. So the essential point is that a two-dimensional epithelial sheet looks in many ways a lot like a two-dimensional foam. So this is an actual foam. This is, these are sort of soap films between air bubbles, and it's put between two parallel glass plates, so it's two-dimensional. And you can see that, it has roughly polygonal things that look like cells. And it's well known that the physics of foams is that every one of these edges has some surface tension. And that is competing with the fact that there's a certain amount of air inside every bubble, which you can't compress forever. And so the tension is sort of trying to shorten the edges, but you can't just shrink everything down to zero because you have the air in there. Our understanding of the way epithelial cell shapes are determined in animals is very similar, with the important difference that the animals are what you might call an active foam. So in the context of statistical physics, what active means is that you are burning ATP to have forces that sort of don't come from trying to minimize an energy. And so for an animal, what that means is if this were an animal cell, it could decide, I want to increase the tension on this edge. I'm going to target some kinase to this edge, which is going to phosphorylate my myosins. The myosins are going to become more active, and suddenly I'm going to pull harder on this edge. So we think of epithelial cell packings as foams, but as active foams which can regulate the tensions on the edges in non-trivial ways. Okay. So what that means is that we have a model in which every cell is a polygon. And inside every cell there's a pressure which is, essentially to keep it near a constant area. You can vary somehow how soft or stiff that is. But more interestingly, there's a tension gamma sitting on every edge. And we can put into our model various ideas about the biology of how that tension gets regulated. So one thing to note is that the edges in general come together in threefold vertices. And you can learn a lot just by thinking about these threefold vertices. So in particular, all of the evidence is that the forces on these vertices are close to balancing in most epithelia. This is basically because cell movements are slow on the timescales of more molecular relaxations, and there's a lot of dissipation and drag in these things. So the vertices aren't moving very fast, which means that to a first approximation, the forces on them have to balance. So then, if you think about, I have three edges coming in that are pulling with three different tensions, and these three tensions have to add up at each vertex. What that means is that you can learn things about the tensions on the edges from looking at the angles between them. So if they all three have the same tension, then I need 120-degree angles, so these three vectors add up to 0. On the other hand, if I see something more like this, with two edges that are close to parallel and a third one that's close to perpendicular, this is telling me that the tensions on the two parallel edges must be much higher than the tension on the third edge, because otherwise they couldn't possibly sum to 0. And so by looking at a picture of the cells and focusing on the angles where edges meet, you can infer things about the tensions. And this is not my work, but there's a fairly sophisticated framework now for doing this on large number of many images of tissues in an automated way, with the right priors to make sure that it doesn't do anything strange, like suddenly decide that the tensions are going to vary by a factor of 1,000 across the cell. So this force inference game is actually quantitative now in a way that I don't particularly make use of. But we can make use of the qualitative part, which is if we blow up some of the 3-cell junctions in my fish images, what you can see is that they are essentially two almost parallel edges vertically and a third edge coming in close to perpendicular. And so what that means is that the tensions on these junctions must be very different. And in fact, in particular, that there's a directional anisotropy, that the vertical ones are under much higher tension than the horizontal ones. All right. So, and this is, in biology terms, what's known as planar polarity. And it's known how you do this. Or at least there are examples of how you do this that are known. So it turns out that this phenomenon of planar cell polarity is quite common in epithelia. I would say it's probably present in most of them, although it hasn't been characterized in all of them, but every time people look, it seems to be there. And the idea is the following. So you have a collection of cells in the plane, and they want to pick out one particular direction as different from the other. So they want to know that vertical is high tension and horizontal is low tension. And in cartoonist fashion, the way they do this is they have two kinds of proteins, A and B, that are membrane-bound proteins. And so they have intracellular domains and extracellular domains. And you set it up so that the intracellular domains really don't like each other. And that means that they will tend to try to segregate to opposite sides of the cell, like this. And this defines a vector, say, from B to A in every cell. So you've lost the rotational symmetry in the cell and picked out a special direction. Then you would also like all of the cells to agree on what the special direction is, and that means that these vectors should talk to each other. And the way that works is that the extracellular domains of A and B like each other. So you have these proteins, and what they're trying to do is segregate to opposite sides of each cell, but have A against B across the junctions. And this, in effect, gives you a little arrow, and for physicists, we think of this as basically a little magnet in every cell, and these arrows are coupled so that they all want to line up. And this picks out a global orientation in the plane. All right. So the mechanics of tensions on edges plus this idea of PCP are the things that go into our fish model. So in a little more detail, the model of planar cell polarity in the fish. So planar cell polarity is very well characterized in some fly systems and in a few vertebrate systems, particularly in fly systems. There are, in fact, multiple known PCP pathways now. We don't really have it well characterized in fish retina. We have identified some of the genes that are involved, and they're sensible genes, but we don't know exactly how it's wired up. So our model is entirely phenomenological. What that means is we think there's PCP, and so we're going to make up the simplest thing that will give PCP and see what that does to observable things like cell shapes, and evaluate whether this is a sensible set of assumptions based on whether it gives sensible predictions for the things we can actually look at. So that means that we only have proteins A and B, and these have a concentration that lives on every edge. And then they talk to each other, and they talk with the interactions I've suggested. So we give them an energy. There's no reason that it has to come from minimizing an energy, but it turns out that you can convince yourself that for the particular case of planar polarity, that's not a very big restriction. That the things that are not variational, that don't come from minimizing energy, only enter at fairly high order in breaking rotational symmetry in the plane. So we can get away with having just an energy, and this energy has quadratic couplings between concentrations, and then it has entropy terms so that none of the concentrations try to go to 0 or infinity on any edge. And these concentration terms are also set up so that the proteins that like each other like to be at high concentrations, and this means all things being equal, you'd rather have high protein concentrations on short edges and low protein concentrations on long edges. And so this means that the PCP sees the cell shape. All right. On the other hand, there's then also a mechanical part of the model. This has the physics I told you about. There are and again, this comes from an energy, and there are reasons why, again, it's moderately sensible to make this come from an energy, even though we don't believe microscopically that's how that happens. And so the energy is that there is a term proportional to the length of every edge, that's the surface tension, and then there's a term that says that every cell has a preferred area, which gives you a tension. So then what does not come from an energy, and is sort of the interesting new part here, is these tensions depend on the concentrations of the PCP proteins on each edge. So as I said, you can regulate the tensions, and the most obvious, the leading term has to be this linear. And so the tensions depend on the concentrations, and specifically based on what we know about the shapes of the fish cells, we think that the presence of these proteins on the edges should lower the tensions. There are relaxational dynamics, non-variational cross-conflict links. What all of this means is that we take derivatives of these energies and we turn this into a whole bunch of ordinary differential equations for the positions of every vertex. And then you go and you try to solve that for positions of every vertex, I should say, and for the concentration of A and B protein on every edge. And so you have, 24 ODs per cell, more or less. I guess I'm not double counting the verse, so more like 18 ODs per cell, and 1,000 cells, and you want to solve all of these coupled equations to see this packing evolve. Okay. So in the spirit that this is a tools and technology seminar, I will say a little bit more than I normally do in this talk about how you actually deal with all these equations. So many coupled ODs is in most cases a done problem from the perspective of numerical analysis. It's been studied for a long time, and solving ordinary differential equations, even lots of them, is something that people do very well. What is hard about these models is that the variables don't remain constant over time, because there can be changes in the topology. So that is, you can have cells that divide and cells that are lost, either to apoptosis or to extrusion. In the fish, the division is pretty negligible, but there are mutants where the extrusion is really important. And so if that happens, suddenly you've lost a cell and you have 3 vertices that merged into one, and you need to update the variables you're keeping track of to lose 2 positions and keep the 3rd position. There are also far more generically things called T1 transitions, in which an edge shrinks to 0 so that I now have 4 edges coming into a vertex instead of 3. And then it resolves itself in the other direction. So essentially, I have 4 cells that have swapped neighbors. And this happens all the time. The mechanics and the energy will want to drive an edge length to 0, and then it will be energetically favorable to pull it out the other way. And every time this happens, again, the variables you have and the couplings between things, so the set of differential equations you want to solve changes. Okay. And so this is what makes simulating these vertex models anything more complicated than sort of taking a standard library ODE solver. The way we deal with this is, this is coded in C++, object-oriented. That per se is not that shocking, but we really make use of the objects in a way that many well-established numerical codes actually don't, because there are, some of the best. Many of you probably know that some of the best libraries for doing some common task basically go back to things written in Fortran 40 years ago, and they still work so well that people still use them. So we, on the other hand, are actually going to use the fact that we have an object-oriented language to have objects that represent vertices, edges, and cells that live in linked lists so that I can easily get rid of some when I need to and easily stick new ones in when I need to. And then every edge is going to have keep track of the protein concentrations on the edges. And the objects are going to keep pointers to their neighbors. So every vertex is going to keep pointers to the edges that come into it. Every edge is going to keep pointer to the neighboring cells. Cell is going to keep pointers towards vertices and so on, so that they can find their neighbors when you need to know about the neighbors to calculate forces. So this is good in that it lets you put in lots of topology changes. I have to say that what is bad about this, which we sort of live with because this is research code, and sometimes you live with, sometimes the problems in the code are less bad than the amount of time it would take you to fix them, is that you have to be really careful every time you change the code, because there are not natural ways to check that you have rearranged the pointers correctly when you merge 2 vertices. And if you get that wrong, it will propagate downstream and make a mess somewhere in a way that's hard to back out. All right. But that said, otherwise you can deal with this. The other thing that this costs you is that it's a lot less efficient to solve these things. So you can, between topology changes, you can use any standard ODE solving algorithm you might want. But it turns out that if you have to stop often enough to check for topology changes, first order Euler is not particularly worse than anything else. And I find this rather disheartening, but it is empirically the case for us and for other people who use these sorts of algorithms. That sort of the overhead to restart fancier things every time you have a topological change negates a lot of their advantages. Okay, so you have it set. Obviously, we have it set up in a modular way so that you can swap in different solvers, and but often we just go with first order Euler because we don't gain that much from not. The biggest computational cost is getting cell areas. So you need to know the cell area to figure out the pressure, and there are formulas for cell area as a function of vertex positions for an arbitrary polygon, but they're computationally expensive, and they're doubly computationally expensive because you have to the vertices, and you have to follow a train of pointers to find everything else that goes into the calculation. So we are dominated by the cell areas in such a way that we can be careless about other things because it's a 1% effect on the cost of calculating the area. the other thing that is not completely trivial is that we have this dynamics of concentrations on the edges, and this can be very stiff. I should possibly stop and ask. Do you all know what stiff means in this context? No. Okay. Right. Small numerical analysis digression. So the standard algorithms solving ordinary differential equations work wonderfully when there's basically only one time scale in the problem. If you have an equation that describes processes happening on two different time scales, so there's something that happens fast and something that happens 1,000 times slower than it, this is called stiff. There's a wide range of time scales. And this introduces potential numerical instabilities very generically in a way that makes it essentially unworkable to use an out-of-the-box render-coded algorithm, say. Or at least in particular, if you want to use an out-of-the-box algorithm, you have to use time steps that are set by the fastest time scale in the problem. And you might really not want to do that if you want to look at evolution on the slow time scale. And so there are very well-established ways to deal with this, which involve using what's called implicit solvers. You pay a computational cost in inverting matrices, but you kill off the numerical instabilities. And so our PCP dynamics can be very stiff. If the concentrations get too large or small on an edge, they want to relax away from that very fast, much faster than the vertex evolution. And this means that you need to use an implicit algorithm. And it turns out that we use a custom mixed implicit-explicit algorithm because in general, implicit algorithms have to invert n by n matrices if you are dealing with NODs. And so if n is 5,000, you don't really want to invert a 5,000 by 5,000 matrix every time step. And we know particular things about the structure of our model that tell us that these matrices are almost diagonal in a particular way. And so we exploit that to develop a particular custom, it's called IMEX discretization, that lets us deal with the PCP dynamics in a way we can live with. We also have to be careful to implement conservation laws for the proteins in a correct way. All right, so that is numerics for the vertex models. Let me remind you then what the model does. So I have PCP proteins, they like short edges, and this means that that the protein sees cell shape. On the other hand, the protein concentrations change tensions, which means that that shape affects concentrations and concentrations affect shapes. And so when these things decide to pick out a special direction, they're going to do it together, and you're going to get one breaking of rotational symmetry. And the idea then is why this would be relevant to the fish in mechanical anisotropy is that this is very much like a magnet. And so most of you will probably at some point done the experiment where you take an iron nail and you rub it with a magnet and it becomes magnetized. So what's going on there is that the iron wants to be a magnet. Its lowest energy state would be that it's always magnetized. But when you take molten iron and you cool it in zero magnetic field, all the atoms in the iron can't all get together and agree on which way their magnetic moment is supposed to point. So you have little domains, depending on the material, hundreds, thousands of atoms, that all feel each other and say, okay, we're going to line up this way. But then over here, some other set of atoms decide they want to line up that way, and then they're frustrated and they can't come to an agreement. And so you have lots of little domains pointing in different directions. And by rubbing it with a strong enough magnet, you reorient these domains so they all point in the same way, and now it's magnetized. The other way to solve that problem is if I were to cool the molten iron in a magnetic field from the outset, then all of the magnetic moments and all the atoms would know which way they're supposed to point. They're supposed to align with the field. And so the thing would be magnetized from the outset. The idea is the same thing is going on here. If I turn on PCP in the fish eye without any signal telling it which way to point, then the cells are going to try to orient themselves in some direction, but they're not all going to agree on the direction. And so you're going to get little pieces of fragments of columns that look like they're trying to line up, as here. But then, over here, some other cells are perversely decided that they want to go that direction instead of diagonally. And where these things match up, you're going to lose the order. On the other hand, if these things turn on PCP in the presence of a pulling stress that tells them this is the good axis, then they will all agree and they will make nice long straight columns. That is the idea. Okay. So, and we can test whether this is plausible in a simulation. So the white cells here are undifferentiated, no PCP. there would really be dividing cells out here, but they're separated by 5 or 6 cell diameters from what we're interested in, and it turns out to be easier to just start with a field of the same number of cells than to constantly have division. And then these, at some point, we're going to by hand take a row of cells and say start differentiating, which means become gray and turn on this polarization and get a vector telling you the direction of the polarization. And all right. Okay, you can see this is what it does. And so this physics is enough to take you from a disordered on-average hexagonal packing to something that's really straight columns lined up. It's not completely perfect at it. So there you can see there's a defect, for example, there. And in fact, there are occasional defects in the fish. But it does pretty well. Equally important, it fails where it's supposed to. If it would advance, we could say that. There we are. Which is that it doesn't work if I don't have the stress anisotropy. So this is everything the same, except that I'm not pulling in the vertical direction. And so you can see that these cells, as they turn on PCP, are trying to line themselves up into columns. And as we go on, you will see that sometimes their attempts lead them to make sort of circular swirls instead of things that are straight. But they don't. And as they grow progressively, they do somewhat better. But still, without something helping them and telling them this is the right way to go, they get confused and they make errors. And they make considerably more errors than when I have a nice global signal telling them which way they're supposed to go. Yeah, and you can see it keeps on evolving, and these errors relax, so on and so on. Here, the big curvy thing. All right, so this works when it's supposed to, and it breaks when it's supposed to. Okay, I don't think you want to hear about pneumatics. So let me, in the final 10 minutes, say why I think this model is not nonsense and how it goes back to how it relates to the actual fish. So it's a general matter of modeling philosophy, right? Biology is very complicated, of course. And so whereas if you come from a physics background, sometimes there are cases where you're just sure the model is right because, a hunk of silicon has silicon atoms and nothing else, and maybe you can't solve the problem of what the silicon atoms do, but everybody agrees on what the problem is. In a fish, we don't know 2/3 of what's there, and that is going to be the case for the foreseeable future. And so even models that are very elegant and clever and seem to explain lots of things could just not be the way the fish decide to do it. And so therefore, there's a much greater premium on checking the model and going back and seeing that it connects to experiments and makes useful predictions. Even then, there's some chance it will turn out to be utterly wrong 5 years from now, but at least it will have advanced your experiments in the meantime and given you a way to think about the thing that's possible. So this means we should try to predict things and we should try to check them. And this is much easier said than done in many cases. So I will tell you some of the things we can check here and some of the things I wish I could check, but we haven't figured out how to do yet. So the first thing is I talked about this idea of that in the absence of a global field, you should still have columns of fragments. And I showed you some pictures. Here's some more pictures of the larva. And again, you can see these here, for example, between these two stars. There are fragments of a column that's trying to line up. This is a lesioned eye that is similarly messed up. And again, you sort of see fragments of columns going one way and fragments of columns going a different way. All right, so these things appear to have some tendency to want to line up in columns. Rods. Okay, so there are rod photoreceptors as well as cone photoreceptors in fish. They insert into the epithelium after the cone pattern has been established. And what's interesting is they systematically insert between columns and not within columns. And so you can see that here, that they're all sitting between columns. The green is the rods, white is now the outline of the cells, and here's the overlay. And all of the green is staying between the columns. And this is what you would expect if the vertical interfaces are high tension, because high tension is the same thing as weak adhesion, and therefore you should put rods there. We also predict, of course, that there's planar polarization, and that means you ought to be able to go find some planar polarized things. And at the time we made this prediction, these hadn't been seen in fish eye. Now several of them have. So the green is crumbs 2b, and specifically it's crumbs 2b in what's called the subapical region. And so you can see that it's primarily along the horizontal interfaces, not along the vertical interfaces. So there is planar polarity. If you are a PCP aficionado, it's interesting that this is Crumb's 2B because it's not classically a PCP protein, but there's a few examples in fly where it is known to be planar polarized, and in planar polarized in cases where there's strong anisotropy in mechanical forces as well as in protein concentrations, as we believe is happening here. So that's kind of nice. So better is that you can kill all the photoreceptors and the retina will regenerate. As I said, this is one reason this is medically interesting. But it will regenerate in a way that's not nice and organized. It sort of, the whole retina says, oh my gosh, I need to regenerate as fast as I can, and so it makes a big mess instead of doing it in this progressive stepwise fashion. But even so, what you can see, so here there are more rods, and that's why there's more blue mush between the cells. But the big cells here are the cone cells. And you can see that they're lining themselves up into line-like sort of fragments of columns still. So clearly these cells have a strong intrinsic tendency to have directional interactions. So that idea, at least from the model, we're fairly confident is good. There are also mutants that do this. So what we would really like to do, of course, is we would like to remove the mechanical anisotropy and see that we break the pattern. So if you do that in the model, just to remind you, what will happen is the following. So here there's mechanical anisotropy and it's making very nice columns. Now I'm going to imagine that I can magically make the annular ligament and thus the mechanical anisotropy go away. And you can see that it expands in one direction. And it's going to continue trying to make a pattern, but you can see that it's going to do rather worse. It's going to start introducing errors, not a ton, but clearly more errors than back there. And then I'm going to put the anisotropy back on, and it's going to recover. And so the prediction is if we could remove the annular ligament, you would see a region that's disordered and then more order. So this is an experiment we've been trying to do for, and by we I mean mostly Pamela, have been trying to do for something like 7 or 8 years now, and the fish really objects very strenuously to any attempt to do this. Okay, and so in particular, you can get rid of the ligament, but the way you have to do it is so violent that the controls also show disruption in the eye, which means it's not a good experiment. Okay, but it turns out that there's a natural version of this that is not as good as our being able to do it in a controlled way, but is still, I think, quite good, which is aging. So this is a 5-week-old fish. This is a standard bright field image. And this is an image in a red fluorescent channel where we have a transgene that's fluorescently labeling the annular ligament. So you can see that this circle sits on top of the eye. So there's this red circle around the eye. And it's very prominent, and there's a strong signal. And if you blow up, this is higher magnification of the ligament and there's lots of red. Okay, so 5 weeks old is a young juvenile fish. It's just barely not quite sexually mature. On the other hand, you can take an 11-month-old fish, and an 11-month-old fish is really pretty elderly. And what it turns out is that the annular ligament degenerates. So this is exactly the same set of pictures, namely the standard light microscopy and then the fluorescent channel. Except that here, where there ought to be a nice red ring around the eye, there's instead just a smidgen of red you can kind of see there, and certainly not a nice strong ring. And again, if you go and do the histology, this correlates, and so there should be ligament tissue there, and there's not. If you look on this side where there's this little red half moon, there's still some ligament tissue that's called the temporal side. Temporal, for reasons I don't know, is the opposite of nasal, so the side away from the nose. And but mostly it's lost the annular ligament. And so we predict that it will also lose the regular packing of cone cells. And this is indeed what happened. So this is not the same fish, but it's a 12-month-old fish, so also elderly. And you can see that mostly the cells are arranged, the cell packing is a big mess. It's certainly not very regular. On the temporal side, where we think that there's still a bit of ligament left, there are still some hints of columnar order, although it's not very good. But basically, the loss of the annular ligament correlates with loss of the crystalline mosaic. So we can't do the experiment we would really like to do, which is manipulate in a controlled way whether or not there's mechanical anisotropy. But the correlative evidence is quite good that both the appearance of this ligament and the disappearance of this ligament in various contexts implies leads to disorder in the cell packing. I should say there are also mutants that misregulate the intraocular pressure, which again misregulates the mechanical environment of the retina, and that also disrupts the cell packing. All right. So I'm going to skip the new directions and go straight to the acknowledgements. Right. So let me come on. Right. So what have I told you about? So I told you a story about retinal cone mosaics and this remarkable fact that they have this crystalline ordering of cells. And I've told you that we have this idea that tissue-level mechanics might be driving this cellular-level regular ordering. I've told you a lot about how we build models, including planar cell polarity and the general class of Vertex models, and shown you some experimental tests that are not slam dunk, but are at least consistent with our story, including the fact there's very clearly a directionality to the interactions and a preference for making column fragments in even the absence of global order, and also the loss of ligament in elderly fish. And okay, I didn't get to chiral symmetry breaking, but this question of how concentrations on edges and cell shape interact is a more general one, and there are more general theoretical things you can ask about. And with that, let me close and acknowledge my collaborators. So as I said, this is all done with the fish lab of Pamela Raymond here at the University of Michigan, we are very grateful to them because I wouldn't know how to keep the fish alive, let alone do immunohistochemistry or something like that. The theory side and the modeling side was started by a really excellent postdoc, Guillaume Salbreux, who actually, this slide is about 6 months out of date. He just moved to the Crick Institute, but so he is now off doing great things on his own. And more recently, some work has been done by students Jeremy Hadjoyo and Meryl Spencer. And as I said, we're also grateful to Pamela, a longstanding collaborator, and of course to the NSF, which gives us money. So thank you for listening. Yes.
You might have mentioned this when you were talking about the stiffness with the PCP dynamics. But what's happening at such different timescales that's causing that stiffness?
So okay, what's causing the stiffness is that you need. So okay, you have interactions, pairwise interactions between proteins that want to put them all on one edge. Okay? And then you also have to have some term that tells you don't let the concentrations go to 0 on any edge, because that becomes pathological. And what that means is you have to sort of have some force that's pushing the concentrations back from 0 that becomes large if you get too close to 0. And so where it gets really stiff is if some concentration tries to get too close to 0, and then it basically gets beaten back on a fast time scale. So you have sort of divergences in the right-hand side of the ODE as you get to 0 that will make it stiff. Yes.
When the larval eyes become an adult eye, it clearly orders. So is that transition from disorder to order due to new cells that grows in this pattern, or is just rearrange of old cells?
No, so it's due to new cells that grow. So in the adult eye, there's a little patch in the center that's the larval remnant that remains disordered because it's the cells that were born when it was a larva, and they had disordered packing, and these things appear not to be able to rearrange themselves substantially once they're fully differentiated and done patterning. So it's the new cells getting added on after it becomes an adult that have a regular mosaic.
Any other questions?
Well, thank you all so much for coming.
Thank you.
Have a good day.